Okay, so the plan now is to have a little discussion around what's transpired so far this morning, which is, my head's spinning, I must say. So if the people who spoke this morning, if they could come up and... Can we get a light on the stage?
I need your mic.
Oh.
What a pity.
So, I mean, I think this morning we've already heard some amazing science, and we've heard some needs, but we've also heard that these things are being met to some degree, and things are going on within existing labs and within existing resources, sometimes at the national level. I mean, the question I would ask the panel, and then also the audience, is: do we need, what else do we need? Do we really need to have something new? Do we need something along the lines of what I was describing in the beginning?
I think we desperately need better tools and infrastructures to solve exactly the problem that you described, that science is very inefficient, and that this type of science uses the kind of technology that other sectors have shown can increase efficiency. There was a long time when people were pushing stock prices up because of the excitement about information technology in Silicon Valley, during which the economy wasn't actually benefiting in productivity from those new technologies, other than the sector that was developing themselves. Subsequently, of course, we now know that most industries are benefiting from information technologies because very solid tools have been developed for their use, and that's what we need. We need better engineered, more reliable tools to make our science more efficient. And you talk about an app store, and the kind of vision I have is very similar. It's sort of an iTunes for multi-scale modeling, that iTunes itself does nothing, but it provides us a common portal to the different data sets and the different tools. What we need is that, and a way of being able to integrate those tools, run the book reader and the music player and the video player simultaneously in novel ways using different types of data. So I think we need software infrastructures of the kind that the NSF has been envisaging, that enables the community to have an app store and an iTunes for multi-scale modeling.
People, I think James would want to say something about that.
No, I think that as a long-term goal that's absolutely right. I think we're a long way from having the understanding of the problems and the infrastructure in a more detailed sense to be able to get there. I think that if you look at an app store, the interoperability is fairly limited. You're interacting with a host operating system, and you have a fairly well-defined set of questions that you're trying to deal with. So you can have a set of APIs for graphics and things like this. Here, we find, for example, there is no way of describing cell behaviors that's a universally agreed upon language. You go inside the cell, there's no way of describing, again, the behaviors of subcellular components that's agreed upon. Until you have a set of encapsulations that are agreed upon, you can't develop a set of APIs that will allow interoperability. And so in some sense, we haven't done the science that's needed yet, or at least as a community come up with a coherent agreement on what is needed at these levels. If you look at the really successful multi-scale integrations, the two examples that are always given are, one, are in genomics, where there are standards, of course, for describing sequences, and therefore you can go from a sequencer all the way through some kind of protein folding or molecular dynamics computation without having to manually reformat, relink, so on. But there you're dealing with a very limited kind of data set. The same thing happens in geographical information systems. That's perhaps the biggest success story. In our case, we don't yet have an agreement on what the core concepts that we want to describe are. And that's why I tried to make the point about having to define the kinds of questions that we're going to be answering, because unfortunately, anytime you do these encapsulations, you are imposing limitations on the kind of questions you can solve. And so I think these very generic platforms are useful, but what we need is, in a sense, that middleware layer, and that requires community agreement on question and approaches and encapsulation.
I think that's actually a very good point. I think there's probably comments from the audience about this. So, Peter, do you?
It's not a comment directly about that. It's more just asking the panel if they can come back to the question that Phil asked at the beginning, which is which is really a social question, how do we change behaviour in the science community so that people actually care about reproducibility? Because, you know, what we're seeing is wonderful science, wonderful tools and so on, but we're not addressing the question, how do you actually encourage people, reward people to care about making their work available in a reproducible form for others to use?
So, Phil.
Yep.
Do the panel want to comment on that? Sorry, before we.
This is a big problem, and I don't think we're going to fundamentally change the paramount importance that publications and the archival journals have in our field in the short term. So I think the answer lies in encouraging the journals and the journal editors, like those of PLOS Computational Biology, past and present, to come up with new ways to encourage and authors to do this, to make these models and data more shareable and more accessible, and that the reward they will get from that is having those papers frequently cited when they're used, just like the PDB.
I will just comment on that in the sense that relating to the Public Library of Science, which is now, you know, a major publisher in this area. PLOS One published over 2,000 papers last month alone, and there is definitely interest within PLOS for elevating the value of data and methods and software and workflows within the context, and essentially making them citable references. Mark and then Terry.
Yeah, so I want to deconstruct and try and reconstruct a little bit. I think James went off in the direction of the challenges of interoperability of simulators and different things that one needs to bring into a simulation environment. If I stack that problem off on the side for the moment, come back, I think, to what Peter was starting to go at, which is reproducibility and sharing, I think. One of the challenges that we put forward, I guess, 8 years ago and got NSF excited about, because it's a problem, is the lack of reproducibility and the lack of current science based on informatics retaining the underlying notion of the scientific method, that is, that you could reproduce work. So that was in the context of a meeting of all folks involved in workflows to ask if the workflow environments that were emerging and some way of dropping codes into them could be used to reinforce the provenance of what's done on data before it goes into a database. You went around the table, there were 20 of us there, from Europe, from the US, from Japan. Nobody had the same understanding of the word workflow. You talked to Miron Livni, it was Dagman running on a single device. So first of all, when we use some of these loaded terms in the context of a meeting like this, we need to make sure we all understand that we had the same meaning. What has been done since is to create things like Kepler, and Taverna does this as well, few other tools do, where at runtime, you have a hierarchical record of exactly what was done, and you can publish that. It's not what's going to go in your paper. You're going to export to the paper what's appropriate for materials and methods, but some other place can retain the details that are required for actual replication on hardware that exists today. The other thing that's been coming of this is this mic doesn't work.
Really?
Everybody can hear
Good to
you.
me.
We could hear you.
The other thing that's been coming of this is a better understanding of what in modern software development methods you use in order to make things run in multiple platforms. So simple services architecture. Even if you've got some big monolithic code, you wrap it and you make it something that drops in. All of the groups that are creating workflows have come to general agreement about what the surfaces of those modules should look like. So at least, you know, the underlayment is there for an iTunes-like definition, an API, if you like, of what would be required. But I don't think that we've defined a set of end-to-end challenges in order to do that in the context of the virtual cell, which I think is the challenge of this meeting.
So I think to kind of get back to this and to address James's point a little bit, I think that's where the benefit of paradigms at work comes in. If we want to sit here and define the entire API for all of integrated biology, we'll never get it built. But if we have an app store model, we don't have to. Each app only needs to export an API that is necessary to use it. And if someone comes along with a better model that requires more, they can extend that API. And those apps that use well-defined APIs and are standardized will be popularly used, and those that don't will be less commonly used. I think Herb Soro had his hand up earlier and probably has a comment on this.
Yes.
Yeah.
So Herbert and then Terry.
Yeah, thanks, Andrew. In fact, I wanted to comment on what you were saying, actually, about the inefficiencies we have today. I mean, I see a lot of resources wasted between NSF, DOE, and NIH in software development. And one of the things that bugs me in the community is that a lot of software that's produced is closed. It's open source, but it's not reusable, or not easily reusable. And it goes back to this point about APIs. Now, the SPML community has a good track record for producing libraries that are reusable. And my own group, we have a whole bunch of libraries that are reusable. In fact, James Glazer's work uses one of those. So I think that's part of the problem, is that people tend to build monolithic, non-reusable code, which is a waste of time in the long run. In the short run, maybe it has benefits, but in the short run, so that's the first point. The other point, we'll go back to reproducibility. I don't understand why people, I mean, we put grant proposals in on reproducibility, and it keeps coming back where reviewers say reproducibility is not an issue in computational science. And I don't understand where they get that from. And of course, at that point, the grant dies, the proposal dies, and you have to wait another 9 months. So this community, I think, hopefully understands there is an issue in reproducibility in computational science. But there are other groups somewhere who don't think there's an issue. And I wish we could get over this issue, this problem. It's been going on for years now. So I completely agree with the problem of reproducibility in computational science.
I wanted to comment a little bit on that, because one of the problems is essentially bootstrapping. The investment required to build reusable code, to build APIs and libraries, is very large. If you are in a new field where the number of users is fairly small, the overhead associated with building good modern code is very high compared to the overhead of building closed code that only you are going to use. Once the community grows, then the advantages of the shareable model are tremendous. But you don't get those network effects without having the platform. You don't build the platform typically until there is a community. And so there is a bootstrapping problem. How do you get some of these things started? SPML has been a wonderful 20-year project. Peter Hunter and CellML, VPH have also been very long times to build the community and to build the infrastructure. And so one of the things is, how can we do the science and the infrastructure building at the same time in a way that is maintainable, instead of having to wait until all the science is done and then doing the infrastructure, or vice versa, having to build all the infrastructure without knowing what the science is that we're going to do. It's a difficult problem.
Terry. Press the bottom of the mic.
Hello? Okay. Turn the discussion from software to data and suggest that we should ask not what data could do for us, but what we could do for data. That is to say, it's been our experience that if you're trying to model a cell or a particular molecule, and you go to literature and you find labs that are studying that cell or that molecule, they've done a lot of measurements, but often they leave out the crucial ones that you need to do the model. And the question is, how can we increase the dialogue? How do we get the feedback loop going so that while they're doing the experiment or before they do the experiment, they have some input from the modelers to know, what's crucial or what's important?
That's exactly what I meant by model-driven experiments, where until you start building a model, you may not understand what data is actually important about a system. We find very often that specifying the model may be enough. You may not actually have to ever run the model, because when you do the specification, you realize that you have lots of molecular data about certain things, and you're missing data that's much more important. And you can't build the model because you're missing key data. Then you know what experiments you have to do. So that's why I say this. You have to close this gap between experiment and simulation. And if you want to use these compressed terminologies like model as data, data as model, those are slogans, but they do have meaning if you unpack them. And I agree completely. We have to be able to pull these things together. One way to do that is to use the same standards to specify model and data. So one of the things we're very interested in is microscopy annotation, not at the subcellular level, but the multicell level, so the 20x level, so that you could take a 2-photon image that's been segmented, annotate it by cell type, and run that as a simulation. Now, we can't do that yet, but we can define the steps that are needed to do that. If you can do that, you have something else that Maciek has emphasized and Jim Sluka have emphasized a lot, which is that nobody's going to use these tools unless they provide immediate, tangible benefits to the people who are using them. Nobody's going to use this because it's virtuous. And if you can define things that provide tools where the microscopist can annotate their data in a way that's useful to them, and they don't care about the modeling, if you happen to have snuck in the information needed to be able to build the model, then you've done something very useful. But you have to have tools that are actually useful and helpful to the people who are going to use them. You can't tell people to use them just because it's virtuous and in the long run it'll be good for the community. And that's another bootstrapping problem. How do you develop tools that provide immediate tangible benefit when the long-term benefit is something that doesn't happen until lots of people have done this? It's a little bit like getting electric cars into the market when you have a gas station-powered car economy. There is a nucleation problem.
A lot of this relates to the general notion that Steve raised, and perhaps in a slightly different context, around lowering the barrier to participation, and which is clearly very important. I think, without a doubt, there's some underlying themes emerging here that we'll be trying to put down so that we form detailed discussions around those this afternoon. But for now, I think it's probably time to have a cup of coffee, don't you think? Yep. So thanks to all the speakers for this morning's session. It was really a great start. Thank you.